Hello everyone, my name is Hussam, and this video we're going to go through our project which was investigating the effect of humidity changes on mass disinfection. My group members are Muhammad Ayub and Abdurrahman Musa, and we were supervised by Dr. Bilal Mansour, Dr. Riza Tafrishi, and Mr. Shamil Abdullah. We will In this video, we will first start off by looking at the purpose of our experiment. Second, we're going to go over the equipment setup and procedure of the experiment. Third, we're gonna portray the graphs present in the results and analyze their significance. Finally, we will end with the concluding remarks, which will contain the summary of our findings and present our reference. So, why did we do this experiment? In 2019, a deadly virus called the coronavirus emerged in Wuhan, China. It quickly spread to the other countries of the world, causing the global pandemic that we know of today. The coronavirus had a huge effect on daily mortality rates, where the last tally made on November 27, 2020 shows that 9,310 people died from the deadly disease. The World Health Organization recommends that we wear masks to prevent the contraction of this deadly disease, and thus it is projected that the compound annual growth rate of the production of masks will double between 2019 and 2023. However, due to the high demand met by the relatively insufficient supply, a lot of people are forced to reuse their old masks, as we can see in this picture. Therefore, we would like to figure out a way on how to disinfect masks before people reuse them using the appropriate humidity and temperature ranges. More accurately, the aim of our experiment would be to reach a medium of simultaneous temperature range between 65 and 80 Celsius and a relative humidity range between 65 and 80%. Now, the yellow line signifies the temperature readings, and we expect it to ramp up from room temperature to the required temperature, which is about 75 or 80 Celsius. And for the humidity, we expect the same thing, as we expect it to increase from 60%, which is room temperature, up until about 80%, and we would like it to remain constant well before the experiment ends. Moving on to the experimental setup, we're going to explain the equipment that we're using in this experiment. So first, we're going to need a closed chamber where the experiment and all measurements are going to be taken. Next, we're going to need a wet tissue that can absorb the water well. Third, we're going to need a heat source to increase the temperature, and that's going to be the VWR hot plate. We will also need the temperature and humidity sensors that are connected inside of the closed chamber, and we need them to be connected to an Arduino that can pass on the analyzed results to a computer where they will be saved as an Excel file. Moving on to the experimental procedure, we're going to explain how we conducted our trials. First, we're going to place a piece of tissue inside of an empty chamber. Next, we're going to add droplets of water on top of that tissue and then close that chamber and place the humidity and temperature sensors as seen. Next, we're going to place the chamber inside of a hot water container at room temperature and then heat that water using our heat source. All the results are going to be read by the Arduino and transferred to Excel. You might be wondering, why do we add water inside of the chamber? It's because we would like the humidity to remain at a constant. However, after we increase the temperature, the humidity of air decreases. But if we add water to the paper tissue, then the evaporation of that water at high temperatures will cause the chamber to have a constant humidity. Determining the mass of the water added will also be an objective for the experiment. For the sake of accuracy, our experiment will have 5 different trials with 5 different masses. The initial masses are shown as 0.3, 0.6, 1.2, 1.5, and 2 grams respectively, and they will be all weighed using an Ohio balance. Now we present the equipment specifications, and as you can see, the most important thing is that both the temperature and humidity sensors have ranges between 60 and 80, which are the experiment ranges. As for the Ohio balance, the maximum it can read is 65 grams. However, the maximum we have for our experiment is only 2 grams, so that means that it's fine, and its accuracy is 0.1 milligram, which is also accurate enough for our experiment. The next part of our video will be talking about the results and discussions of our trials. All 5 trials exhibited similar behavior between temperature and humidity. As you can see, the temperature ramp was increasing according to our desire. However, it did not remain at a constant like we are doing in green right now. This is a source of error in our experiment. All humidity graphs shared the same 3 phases. The first phase was an increase from room condition to almost 90% humidity due to the evaporation of the water added to the tissue. Phase 2 of all humidity trials showed that they remained constant and attained equilibrium at the evaporation level showed. The third phase of all humidity trials showed that the decreasing amount of water due to evaporation and the relatively constant temperature causes the humidity to decrease as shown. After phase 3 is over, the amount of water inside the chamber is reduced to 0, and thus the tissue does not affect the humidity whatsoever. The remaining of the graph just shows the inverse relationship between temperature and humidity. To evaluate our experiment, we should have the percentage of error, and to calculate the percentage of error, we should have both the theoretical and experimental values of the masses of water added. These values can be obtained from both the actual volume of water added through the syringe and through the saturation temperature and pressure of our chamber at equilibrium, which can be found out from this equation. Note that our percent of error is relatively high since our equilibrium did not last for long and that we had to consider the intervals of which no water was present during the experiment. To conclude our experiment, the conditions needed to be met were nearly achieved. However, the equilibrium phase did not last very long in each trial, and the different temperatures makes it hard to compare quantitatively between different masses. This leaves us with no choice but to deem our experiment inconclusive as to which we can actually reach equilibrium for a very long time in the specific amount of temperature required. As a recommendation for future experiments, we propose that all trials be held simultaneously in the same water container. This way, the temperature measurements will be the same, and the humidity measurements between different trials and different masses can be compared, thus reaching a definitive conclusion about the certainty of the experiment.